Hi, this is Esther Caparrós from Miguel Hernández University in the area of immunology, and today we are going to talk about the regulation of the immune system by cannabinoids, inflammation and infection. First of all, just to remind you that the innate and adaptive immune system have two different branches. The first one is the innate, and it has all together cellular and humoral components, and the adaptive immune response is the specific one, and as well it has cellular and also humoral components. So talking about the innate immune components, we have the chemical and physical barriers such as epithelium and antimicrobial molecules produced. Also we have the phagocytic cells like neutrophils, monocytes and macrophages, and we are going to talk about these two cells a lot during the lesson. Also we have the NK cells, that is the acronym for natural killer. Also, we have blood circulating proteins such as complement and other inflammatory mediators. And finally, we also have cytokines, that are the proteins in charge of the regulation and coordination of different immune cell actions. Regarding the adaptive immune components, we have the T cells and also the B cells. And these cells are in charge of producing antibodies, B cells, and T cells, the helper and cytotoxic cells, are in charge of the cellular immunity. So altogether these mechanisms work just to eliminate the pathogen that can be present in our organism in one moment. So these two responses act chronologically, one after the other. First of all, the innate immunity acts as with the epithelial barriers, with non-specific barriers and non-specific cells that can phagocytose and eliminate the pathogen in the first 10, 12, 24 hours. And then afterwards the adaptive immunity is going to participate in the response and to produce a specific response directed to the pathogen. So these adaptive immune components, mainly in this image the T cells, have to recognize the pathogen specifically in the context of an antigen-presenting cell, such as a dendritic cell, and then they can proliferate, differentiate, and give rise to a specific pathogen population, so that they can work in the effector elimination of the pathogen and establishment of the final homeostasis of the system. So, talking about the T helper response, the cells that have to recognize the specific peptide of the pathogen in the context of an antigen presenting cell, in this case the dendritic cell, as we were talking previously. They can give rise to different populations such as Th1 cells, Th2, Treg cells, and Th17. So as you can see here, these are the main activities that these populations can do. For example, Th1 cells can fight intercellular infection, virus and bacteria. Th2 cells work together in order to eliminate parasites and also in the allergenic response, in the hypersensitivity responses. And Treg cells are in charge of the homeostatic response. They can suppress the responses of the other cells. And finally, the Th17 cell population that are mainly activated when an extracellular infection is in our organism, and this can be bacteria or fungi. The cytotoxic T cell response, when it is activated, the name of the cell is CTL. So this response is very effective. They eliminate target cells. These are cells that are infected by virus or cells that are tumoral, transformative. So they can directly eliminate these cells in a close membrane response. And the objective of this response is the final degradation of the membrane and the apoptotic cell death of the target cell. So, in general, main differences between innate and adaptive immune system are these one present in this table very simply. In a simplistic way, we can say that innate immunity is an antigen-dependent response, while adaptive immunity is an antigen-dependent response. While innate immunity exerts an immediate response, the adaptive immunity is a non-immediate response. Also, the innate immunity doesn't give rise, to potential memory, while the adaptive immunity gives rise to an immunological cell memory, and very effective one. So when we have a look at the different phases of the adaptive immunity, we can see that, first of all, our immune system has to recognize the pathogen. They specifically recognize the pathogen. Then they are cloned, differentiated to give rise to a phase of activation of all the lymphocytes. Then the elimination of the antigen is very important by the humoral or cell-mediated immunity. And when everything is solved, we can reach the homeostatic phase with the contraction of all the response and the apoptosis of all cells that we don't need anymore, and the well-established surviving memory cells in the last phase, core memory phase. And in this phase, as we can see by the x-axis, days after antigen exposure can be achieved. The memory cells can be achieved after 20-24 days after the initial entrance of the pathogen. So this is the representation of the phases of the adaptive immunity. So what are the characteristics of the adaptive immune response? Well, the main features are represented in this table. We can see that this adaptive response is specific. So we ensure that the immune responses are precisely targeted to microbial pathogens. This adaptive immune response is diverse. So this ability enables the immune system to respond to a large variety of antigens. It has memory, as we have talked previously, that leads to enhanced responses to repeated exposures to the same antigens. We also can observe a clonal expansion, and this increases the number of antigen-specific lymphocytes from a small number of naive lymphocytes. This is a well-specialized response that generates responses that are optimal for defense against different types of microbes. And also the adaptive immune response has the ability to contract and reach the homeostasis. So this allows the immune system to respond to newly encountered antigens. And the most important thing of all is that we don't have a reaction against ourselves. So this is another ability, non-reactivity to self. This prevents injury to the host during responses to foreign antigens. So this response is orchestrated, and we can have different exposures to the same antigen or to new antigens together with an old antigen, as we can see here. For example, we can make a response of a primary phase, such as the one that we observe here. This is like the representation that we have seen before. So we can have the exposure to a first infection and give rise to a very potent response and generate different memory cells. And if we have a repeated infection with the same antigen that previously we have previously been exposed, and then we have another higher response, secondary antibody response, that will be quicker and higher. And this is very important because memory cells are there. We have them from previously exposure, so now we will achieve a higher and quicker response. So finally we will get different memory B cell populations, also T cells, but in this image we can only see the B cell response that will be memory cell response in charge of maintaining this specificity a long time. It is very important in the immune system to know that CD is the cluster of differentiation and is the markers that we assign to different molecules that have a role in the immune system response. So CD markers are membrane markers that allow differentiation of cells. We can distinguish among different cells. These are molecules that allow interaction and addition between cells, and also they are molecules that can transduce signals into the cell. So if we have a look at this table, we can see that all leukocytes, for example, are positive for CD45. Polymorphonuclear cells are leukocytes, so they have the CD45, but they also express CD15, so they are positive for both markers. If we have a look at monocytes, we can observe the same thing. They are leukocytes, but they express another marker that is CD14. Also, when we have a look at T lymphocytes, as they are leukocytes, they express CD45, but they also express CD3. Again, if we have a look at this subpopulation of T lymphocytes, we can see that they are CD45, also CD3 because they are T lymphocytes, but also they express another marker which is CD4. In the same way, T cytotoxic lymphocytes are leukocytes, are CD3 positive and also CD8 positive. So these markers allow us to distinguish among these different cell populations. B lymphocytes also are leukocytes, but they express another two markers, CD19 and 20, that are representative of this population. And also NK cells express this CD45, they are leukocytes, but they express another two markers, CD16 and CD56. So these markers allow us to distinguish among these different cell populations. So, what is inflammation? Inflammation is the response that is triggered in the infection focus as a consequence of the recognition of pathogens. Aim at controlling the infection when the adaptive immune response develops. So, let's have a look at this. The 4 cardinal signs of inflammation were described by Paracelsus and are blush, which is red coloring of the skin or an organ, tumor, that is the swelling, heat, and pain. Also, Galen added the 5th sign, which is loss of function. So these are the 5 characteristics that are defining inflammation. What are the events that are induced during inflammation? Different events happen during inflammation in the blood vessels. For example, an increase in vessel diameter that allows fluids and cells to reach the tissue where they want to get and to fight the pathogen. Also for this, the blood vessels will be expressing additional molecules, so we can observe an increase of these molecules, and also a chemiotaxis of the cells. The cells are moving to that area where the pathogen is proliferating, and the cells are generating an inflammatory microenvironment. So during the inflammation, we can observe that this can be acute. In the acute inflammation, we observe neutrophil recruitment, chemical mediators, and this inflammation can progress to chronic inflammation, but also it can be solved. Sometimes this solution is not optimal, and the tissue lost its function, and then we can observe fibrosis. Fibrosis is completely lost, the tissue integrity, is completely lost, and the function, that's the most important thing, function is lost. So, which cells are in charge of doing this? Well, different cells of the immune system, of course, are in charge of this, and chronically they begin with the neutrophils reaching the tissue, and then monocytes, and finally lymphocytes. Among these cells, we can observe different events such as activation of mast cells and also basophils that can reach the tissue. Also, platelets are activated, and coagulation can be raised in the tissue, and also another innate system mechanisms such as complement activation or NK cells activity. Finally, in the end, T and B lymphocytes can reach the tissue and help fight the infection. When inflammation finishes, it starts the repairing process restarts. So, if destruction of parenchyma and stroma cells is happening, such as in the process of inflammation, the repair process begins 24 hours after the injury. It is repaired by angiogenesis, migration and proliferation of fibroblasts, extracellular matrix deposit, and tissue remodeling. The tissue is going to reorganize, and everything will be well established if it is solved successfully. But if there can be different problems that don't allow the tissue to recover, and this is the fibrosis situation as we were talking previously. So cells have to communicate between them, and they do it thanks to proteins secreted by different cells in the innate and the adaptive immunity, and this is in response to antigen. So cytokines are proteins that are secreted and that regulate the duration and amplitude of the immune response. They help to recruit cells to conflict zones, areas, that is inflammation, and they can induce the generation of new cells from hematopoietic precursors also, not only the communication but also the generation of immune system cells. Chemokines are also cytokines, are in the group of cytokines, and they stimulate the movement of leukocytes and regulate migration from blood tissues or intervene in the tissue organization. No? In this case, we can observe that cells can move from one side to the other thanks to chemokines. So in places where chemokines are in high concentration, our cells having the receptor for these cytokines, for these chemokines, can move directly to that area of higher concentration of chemokines. More molecules that we have in our immune system, and also not only in the innate but also in some cells of the adaptive, but mostly in the innate cells, are the this is moving forward, and here we are. So we were talking about TLRs. These are toll-like receptors, and these are receptors that are expressed in different cells of the innate immune system, mostly in macrophages, neutrophils, and monocytes. What are these TLRs? These are molecules that are in the membrane of these cell populations, but not only in the extracellular membrane, they also are present in endosomes inside the cells. So if we have a look at this image, we can see that they can heteromerize or be alone in the membrane. So they can activate intercellular cascades for phagocytosis, cellular activation, and production of pro-inflammatory cytokines such as interleukin-1, 6, 8, 12, TNF, and interferon, and induction of the respiratory burst, which is the microbicidal pathway. So let's have a look at one of the most important TLRs, which is TLR4, which is the receptor for LPS, lipopolysaccharide, present in gram-negative bacteria. So it is in the membrane of the cells, but we will see that it's also in membrane of another very important population, which is the microglia. So, what's the immunomodulatory activity of endocannabinoids? Well, in this course, you have had the opportunity to see which is the endocannabinoid system. I'm not going to stop in this, but in this image, we can see that CB1 and CB2 are main cannabinoid receptors, and ligands are phytocannabinoids, our endocannabinoids, and also we have synthetic cannabinoids that can be bound to these receptors. The most important thing is that these two receptors are present in lots of different immune system cells. What are the functions there? Let's see. If we have a look at the localization of CB1 and CB2, we can observe that mainly expressed on immune system cells. So this is very important because these receptors have a function in these cells, and we are going to see deeper which is the effect, or why are they present in these cells? Well, this is a very important clue, is the expression of this receptor, this CB2, in immune system cells. Let's see, what are the functions? Well, it has immunological properties. As you can see here, it can regulate addition, regulate T-cell differentiation, and a lot of different pathways. Let's see. In this image, we can see that CB2 is the receptor that is mainly expressed in immune system cells. Polymorphonuclear B cells, monocytes, this receptor is present in these cells. As we can see here, in spleen, in tonsils, this receptor is expressed, but we will see that not only in these places. As we can see here, CB2 is expressed in cells of the immune system, including microglia, macrophages of the central nervous system. From the highest to the lowest expression, we can see here that lymphocytes, macrophages, monocytes, NK cells, and polynuclear cells are the most less present cells of this CB2. So, this is an example of the microglia. In this image, we can see microglia here. The endocannabinoids produced and the CB2 expression in this. Also, we can observe in the postsynaptic neuron that CB2 is expressed. And what is the function of CB2? Well, there is a lot of information still to be known, but up to date, we know that the main transduction signaling of the CB2 receptor is directed to the suppression of the inflammatory response. So it's a very important anti-inflammatory mediator, receptor mediator, and we are going to see it in different situations, cells, tissues, and illnesses. So in this image, for example, in B, we can observe the intracellular pathways activated by ligand binding to the receptor, such as this MAP kinase, AKT pathways that give rise to the reduction of gene expression of inflammatory mediators. This is very important function of this CB2 receptor in immune system cells. Well, this is a summary also, same direction of the previous slide. We can observe here that CB2 can activate different pathways, but the most important thing is that they are going to regulate in order to suppress inflammatory response. And what effect does the activation of endocannabinoid receptors have on microglia, which is the central nervous system macrophages? Well, as we can see in this image, our microglia have different states. The resting state is very similar to like a tree with the branches, and when microglia is activated, it changes from a very branched state to a more reducing branches state. And then when they are producing cytokine, they are very active, they are very more rounded and less branched. So these cells contribute to maintaining homeostasis and are characterized by their plasticity. This is very important for microglia. They are very plastic cells. They are in charge of eliminating neurons and other dead cells, and and they produce different factors in the tissue. These factors are very different, so depending on the activation that the cell has, they can produce different mediators. They can produce pro-inflammatory mediators, and these microglia give rise to a more M1 microglia, which is a profile of pro-inflammatory cell. And another option for this microglia is a more reparatory profile, that is microglia called M2, which is a more anti-inflammatory one. And if we insert in this image the presence of the receptor of the endocannabinoid system, we can observe that the more reparatory microglia, the more anti-inflammatory, has the more expression of CB2, the receptor that is in charge of giving an immune suppressant response. So these cells produce, you can have a look at in here, they produce IL-10, that is the immune suppressive cytokine for the immune system cells. So we can see here again that CB2 is in charge of suppressing the immune response, in this case expressed in the microglia. So in this image we go to a deeper differentiation of microglia cells. We observe the non-activated basal condition of the microglia in here, then an M1 phenotype, that is the pro-inflammatory, and in this case we observe that CB2 is downregulated, and when the microglia is stimulated to a more reparatory state, more stable, CB2 is already expressed in both this subclassification of M2 phenotype. So this is very important in the maintaining of the homeostasis and the repairing phenotype. So can we change these microglia states? Well, this is the more desirable thing, that if we have a pro-inflammatory microglia, that we could change this to an anti-inflammatory and repairing microglia. So CB2 is a very good receptor that can be used, that can be considered as a target of the immune suppression in different pathologies or illnesses. So we can see here in this image that the M1 profile, the pro-inflammatory profile, can damage neurons, and the more repairing phenotype, as we can see here in the healthy neuron, is promoted by the M2 phenotype of the microglia. So if we have a look at the innate immune system and the adaptive immune system, these cells were thought to be always outside the central nervous system, but years ago one group defined the possibility of the incoming of these cells into the central nervous system. So they come past the blood-brain barrier and have an activity inside the central nervous system. And this is very important for the knowledge on different illnesses that have a target in the central nervous system. And what is the effect of these cells there? Well, as we can see here, the effect of the activation of CB2 that is the most interesting receptor in the immune system, have different implications in different cells. For example, in lymphocytes, CB1 and CB2 activity is reduced for IL-2 production. I mean, IL-2 is a very important cytokine for proliferation of T cells, and the expression, the presence of these receptors are indicating that cells are not going to divide and proliferate as well as if the receptor was not present in these cells. As we can see in the second point, CB2 is involved in the suppression of T cell activation, and this is one of the reasons. Also, it is important for the suppression if one cell is proliferating. The importance of CB2 in this cell is that the ligand will achieve the stop in this cell proliferation. Also, for example, in the case of neutrophils, that are very important cells in fighting of infection in first hours, endocannabinoid activity reduces its mobilization to sites of inflammation. This is very important, not in the case of pathogen fight, but in the case of chronic inflammatory responses. Imagine that the neutrophils are promoting this inflammation in illness, in disease situation, and that we can stop this inflammation with CB2 or endocannabinoid system in this sense. So that's the importance of this receptor here. In macrophages also, as you can see, they can have that immune suppression as we have been talking about. And in dendritic cells, for example, the activity of CB2 implies dendritic cell blocking of migration, and this is also important because dendritic cells stimulate T cells. So if dendritic cells are stopped, they don't migrate, they won't be activating lymphocytes, and this is very important. So in this review, we can see that endocannabinoids are in charge of immune regulation and immunopathologies, and this is very, very relevant review in the case of the immune system and the effects of the endocannabinoid system. On the left, we can observe effects of anandamide, and on the right, we can see the effects of 2-AG. And all the effects in all cells represented here, main of the effects are directed to blocking of the immune response. And this is very, very important for the regulation of these cells, as you can see. So, for example, if we have a look at the collaboration and effect of TLR responses that are the ones activated by toll-like receptors in different cells, you can see that, for example, in alveolar macrophages present in lung, when they are activated by LPS, we can observe that the cannabinoid system allows a reduction of inflammatory cytokines. Also in monocytes and different kinds of monocytes and in mononuclear cells that are circulating in blood. So, cannabinoid interacts in reducing the activation of the immune system. We can see here that in other cells such as microglia, dendritic or other cell lines, the effect is very, very similar. So, our pathogen-associated molecular patterns or damage-associated molecular patterns generate dangerous signals that are received by TLRs, and the response can be the increase of CB2 receptors, and this gives rise to the immune suppression, or the response can be the downregulation of CB2 receptors, and the result of this can be the inflammation. So the regulation of the response is not that simple, it's very complicated, and as we can see here, the exposure to cannabinoids, the activation of the receptor of cannabinoids can give rise to the production of different microRNAs, different molecules that control gene expression, but as a whole, different amount of microRNAs, as you can see here, can act in collaboration in order to give rise to an anti-inflammatory effect. So deeper analysis of these microRNAs and their work have to be performed in order to know how everything is working to give rise to this anti-inflammatory effect. So as an example, the cannabinoid receptors and the immune system cells, in this example we can see monocyte dendritic cells, and if you have a look at the graphs in here, you can see that in control, if we have look at this line, which marks the negative and the positive expression, well, if we have a look at the control and we have a look at the addition of tetrahydrocannabidiol, we can observe that this ligand can make a reduction of all the molecules that can be considered as pro-inflammatory in the cells. So also in this molecule we can see a reduction of the expression when we are increasing this ligand addition to the cells. So same thing here with T cells. So we can see in this image that this anti-inflammatory profile is real, and we can see it in vitro with these monocyte and dendritic cells. When there is an infection, these cells are going to migrate from blood to the tissue. So if these cells have the expression of cannabinoid receptors, they can be promoting the control of the inflammatory response tissue, specifically when they are moved over there. One example of the infection issue is the sepsis. In sepsis, we have dysregulation of the response. We have an increase of bacteria, an increase of circulating bacteria that can make a very high damage to the tissue. But can we differentiate sepsis from another situation? Yes, we can modulate sepsis like reducing the presence of bacteria with antibiotics, but this is a clinical decision. But also the research is directed now to the overexpression of the endocannabinoid receptors in order to promote the anti-inflammatory response that we desire after a septicemia event. So can we achieve this? Yes, we can with these ligands of the cannabinoid system, for example, CBD and THC. These are ligands for the CB1, CB2, and the effects that we obtained after these ligands is the reduction of all the inflammatory responses, as we can see here in this line, in these publications. So, is the cannabinoid receptors signaling system regulation a potential therapeutic target intervention? Yes, it is. For example, if we have a look at this paper, we can see here that the importance of these receptors in pain is well established, but we need more research in this. But you can see here that CB2 receptor was shown to have an increased immunomodulatory response. This is important. Also, in cancer, we will be observing in following lessons that it has an anti-inflammatory, anti-proliferative, pro-apoptotic, anti-invasive, and anti-metastatic effect. Also, in the hepatic metabolism, CB2 has a protective action in the phenomenon induced by ischemia reperfusion injury. In the gastrointestinal system, also CB1 also inhibit the pro-inflammatory and pro-oxidative activities, especially for the colon. In the cardiovascular system, CB2 reduces inflammation specifically related to atheromatoous plaques and reduces thrombosis risk. And also in the immune system, as we have seen, the effect is absolutely anti-inflammatory. So, an example of infection is the Staphylococcus aureus. It's an infection of epithelial surfaces that can give rise to pneumonia and meningitis and septicemia. So, as you can see here, pathways activated by Staphylococcus, the toxins of Staphylococcus, are of production of cytokines and inflammatory responses. These authors observed that if we culture Staphylococcus in a plate, we observe the proliferation of these bacteria. But when it is cultured in a media with endocannabinoids, they can't proliferate as much as they proliferate without any endocannabinoids. This is important because the formation of the biofilm is reduced. So can we control this proliferation of bacteria? It seems that the works that are being published point to this thing. And not only for bacteria, but also for virus. As here we can see, the agonist of the CB2 can give rise to anti-inflammatory factors, and this is what we were seeing in previous slides, and this block inflammation. And this can be controlling disease progression in virus infection. Very important. For HIV, if we have a look at this graph here, we can see that endocannabinoids can suppress pro-inflammatory cytokine production and increase anti-inflammatory cytokines in cytotoxicity produced by the virus. But though we desire a pro-inflammatory response, because this is not good for us, it is important to maintain it in order to clean up the virus. So we have to be careful with the antiviral response or control by the cannabinoids, because we want a response, but we have to control it. So this is just a new target point that can be observed in different infections. So it has been seen that CD2 increases significantly in the white matter in HIV encephalitis. So if this receptor is increasing, it's a good target to go to for the control of the infection. Okay, but we have to be careful with these receptors in the infection when a virus is present. So, but there are nice publications that summarize different targets in viral infection that can be considered in order to be effective in the control of the inflammation when a virus is present. So for this lesson, and to conclude, I want to tell you that the immune system is responsible for the inflammatory response and tissue repair. It has receptors for the endocannabinoid system, different expression according to cell type. Microglia, which is the cell population of macrophages present in central nervous system, respond to variation in endocannabinoid levels, pro-inflammatory or anti-inflammatory responses. The immune system cells that participate in the antibacterial and antiviral response can modulate their activity and response through the endocannabinoid system. Thanks for your attention.